This presentation contains our discussion on the clinically significant Vibrio species, plus Aeromonas, Campylobacter, and Helicobacter. Here is the outline of our discussion. Ako rin po yung gumawa ng voice-guided presentation ng Chapter 20 sa Canvas module natin. And yun po yung hindi pa revised na version nitong presentation na to. Kasi nakasama pa po doon yung Plasmonas shigelloides, which we've already talked about during our discussion on Enterobacteriaceae, di ba po? Because it was only a former member of Vibrionaceae, but it has been reclassified as an actual member of Enterobacteriaceae. So today, hindi na po kasama sa discussion natin yung Plasmonas shigelloides. Let us begin with Vibrio, known as the coma-shaped bacillus or the curved bacillus. Vibrio species belong to the family Vibrionaceae, and they are distributed worldwide in various aquatic environments ranging from freshwater to saltwater. So mamaya po makikita natin kung alin specifically yung mga species that prefer saltwater or those that prefer to be in freshwater. So they have been observed to prefer temperatures above 20 degrees Celsius. So they like it a bit more warm. Okay, so they are more easily isolated during the summer, but their numbers would decline during winter, and most of them are found only among the sediments at the bottom of whichever body of water they may be inhabiting. So anyway, since wala pong winter sa Pilipinas, we can't really relate. All right, so they could also cause a wide variety of infections, from gastrointestinal diseases to necrotizing fasciitis to systemic infections like septicemia. There are 4 commonly isolated species, and these are Vibrio cholerae, both the serogroups O1 and non-O1, which we'll get to in a short while. Tapos po kasama din dito yung Vibrio parahemolyticus, vulnificus, and alginolyticus. So to aid in the proper diagnosis of infections caused by Vibrio species, aside from laboratory findings, a good medical history is also important. So example, we need to know how the patient acquired the infection. So kung via ingestion ba or through occupational exposure, because these information would help us narrow down kung ano po yung mismong species na nag-cause ng infection, since different species could also be acquired in different ways and could cause different types of infections. So some questions we might ask for gastrointestinal symptoms would include, did the patient recently consume raw or improperly prepared seafood? Tapos in cases naman po of wound infections, so we might ask, was the trauma acquired while the patient was in contact with either fresh or marine water or any associated products? Pwede rin naman po, did they already have an open wound when they were in that area kung saan po possibly na-acquire nila yung infection? So yun po, those are the things that the physician has to consider when ruling out infections caused by Vibrio species. This one is taken directly from our reference textbook. So these are a list of clinical infections caused by Vibrio species. So notice that Vibrio cholerae O1 can cause a variety of infections such as cholera, wound infections, gastroenteritis, and bacteremia. The same is true for Vibrio alginolyticus. So medyo mahaba rin po yung listahan ng mga infections that may be caused by this species. So it has been isolated in localized infections such as wound infections, ear infections, conjunctivitis, and ayan po, these are most likely acquired from swimming in colonized bodies of water. Alright, so Vibrio alginolyticus has even been documented to cause respiratory illness and bacteremia. Alright, so for all species of Vibrio, their microscopic morphology is the same. So hindi na po natin paulit-ulit na ipapakita yung microscopic morphology ng bawat species as we go through because they all look like this. Okay, so onto the general characteristics or descriptions. Vibrio species are gram-negative, non-spore-forming bacilli, and they are motile either via a polar sheathed flagella, usually seen if the colonies are grown on a broth medium, or they may also have peritrichous flagella if the isolates have been grown on solid media. Alright? So ulitin po natin yun, no? Kung yung organisms po is we would keep them and we would cultivate them in a broth media, dun po natin pwedeng ma-observe yung polar sheathed flagella nila. But if we were to grow them in a solid culture media or a solid agar, then most likely dun po natin makikita yung peritrichous flagella nila. Alright, so Vibrio had always been described as a curved gram-negative bacilli, although this morphology is only seen in the initial gram stain of clinical samples as shown here on the photo at the top right. So ito po, kung manggagaling daw po diretso from the clinical sample yung organism na i-observe natin microscopically, ayan, makikita po natin yung characteristic na curved appearance nila. But sometimes they might also appear less curved, just like these ones at the bottom right. Okay, so minsan po baka example ng galing na sa culture or na subculture na natin yung organism before we observe them under the microscope. Ayan po, pwedeng mag-iba yung itsura nila or they might appear less curved. Aside from being catalase and oxidase positive, Vibrio species are also identified by their susceptibility to the vibriostatic compound, or the O129. So as shown here in this picture, a resistant organism can be observed on the left. So kung makikita po natin, wala, kahit even just a small zone of inhibition, ayan po, none of that was formed or was observed. So this one is a resistant organism. And then here on the right, ayan po, compared to this plate on the right, there was a clear zone of inhibition around the disc, which tells us that this organism is susceptible to the vibriostatic agent, and most likely this is a Vibrio species. So madali lang naman po maintindihan or madaling matandaan based sa pangalan nila, di ba? Vibrio species are susceptible to the vibriostatic compound. Alright, so ito pong susceptibility or resistance nila to the vibriostatic compound can help us in differentiating them from other similar organisms like Aeromonas, Campylobacter, and Pleishiomonas. Kasi po, those previously mentioned genera, marami rin po kasi silang similarities with Vibrio species, which is why we have to make use of other biochemical tests or other physiologic characteristics para po ma-differentiate natin sila. And ayan, this is one of those. So again, Vibrio species are susceptible to the vibriostatic compound. Now, as mentioned earlier, Vibrio cholerae has subgroups. So there are actually 3, and these are Vibrio cholerae O1, Vibrio cholerae O139, and Vibrio cholerae non-O1. And all of these above mentioned subgroups share this same flagellar antigenic structure. So kahit magkaiba po sila ng somatic O antigen, as we can see by their names, ayan po, magkakaparehas naman sila ng flagellar antigen. Now, itong naka-highlight po, focus tayo dito sa Vibrio cholerae subgroup O1. This one is further divided into 3 more groups based on its somatic O antigen. So, ang different subgroups po nito is, ayan po, yung tatlo. So, its different serotypes are Ogawa, Inaba, and Hikojima. So, ulitin po natin, Vibrio cholerae has different subgroups, which is Vibrio cholerae O1, O139, and the non-O1 subgroup. Tapos po yung Vibrio cholerae O1, it's also further divided into 3 serotypes, which is the Ogawa, Inaba, and Hikodima. Ayan. So this is based on the differences in their somatic O antigen. Okay? So wag po mako-confuse kasi this is even more, or Vibrio cholerae O1 is even further divided. Kasi maliban po sa mga serotypes, meron din po siyang biogroups. So the two biogroups of Vibrio cholerae O1 are the classic and the LTOR. So Vibrio cholerae LTOR has been the predominant biogroup involved in previous pandemics. So there was a time in history kung saan nagkaroon din po ng pandemic ng cholera. Although there have been re-emerging occurrence of the classical biogroup. Okay, so ayan po, for us to be able to determine if yung isolate nga po ba is LTOR, ayan, we have to check if they are Voges-Proskauer positive, they can hemolyze erythrocytes, or ayan, sila po ay hemolytic, they may be inhibited by polymyxin, and they have the ability to agglutinate chicken red blood cells. Okay, so these are most likely the biochemical tests or the tests that we can perform or the characteristics that we may observe para po ma-differentiate natin yung LTOR versus the classic Vibrio cholerae O1. Okay? So again, ulitin po natin, Vibrio cholerae has 3 serotypes. Yung serotypes niya are yung tatlong Japanese sounding names, which is Ogawa, Inaba, and Hikodima. Tapos, Vibrio cholerae O1 also has 2 different biogroups, which is the classic and LTOR. Ayan. With regards naman po to epidemics, ayan, Vibrio cholerae O1 and O139 have usually been isolated. Let us now discuss the virulence factor of Vibrio cholerae, which is the cholera toxin or choleragen. So choleragen is an enterotoxin consisting of 2 A subunits which are responsible for toxicity, as we can see here in this illustration. So ayan po, diba? Merong A1 and A2 subunits. So together, sila po yung parang pinaka-in charge with the toxicity of the choleragen. And then they also have 5 B subunits which are responsible for binding. So again, as we can see here in this illustration, meron pong 5 B subunits surrounding the 2 A subunits. So how would this work? Ano po ang mechanism of action ng choleragen? So first step is that once the bacterial cell releases the cholera toxin, any of the 5 B subunits will bind to the host cell to the GM1 ganglioside receptor on the surface of the cell. Alright? Ulitin natin yung part na yun, no? After Vibrio cholerae has entered the host, hahanapin nito ngayon yung appropriate host cell to infect. Okay? And they would do this by finding the GM1 ganglioside receptor. So parang ito yung parang entry point nung toxin dun sa cell, or parang ito yung sign nung toxin na nahanap niya na yung appropriate cell to infect or to act upon. Alright? So what would happen is the cholera toxin, or yung cholera toxin or yung choleragen, it would bind to the GM1 ganglioside receptor. So what happens next is the A2 subunit will then facilitate the entry of the A1 subunit into the cell. So kumbaga, yung A2 subunit po, yung tagabukas ng pinto, para makapasok yung A1 subunit. And this A1 subunit, once inside of the cell, this would stimulate the production of adenylate cyclase. Alright, so pagkapasok po nung A subunits doon sa loob ng cell, magsisimula na yung trabaho ni A1 subunit. So ang trabaho po ni A1 subunit is to increase the activity of adenylate cyclase. Alright, so now the increased activity of adenylate cyclase would lead to the accumulation of cyclic adenosine monophosphate or CAMP. So ang magiging consequence po ng pagtaas ng activity ng adenylate cyclase is dadami rin po yung cyclic adenosine monophosphate sa loob ng cell. So high concentrations of cyclic adenosine monophosphate would cause hypersecretion of electrolytes and water to the outside of the cell towards the intestinal lumen, hence the watery diarrhea. Alright, so ulitin po natin yung part na yun. Ang gagawin ni A1 subunit is i-increase niyo po yung adenylate cyclase activity. The consequence of this would increase the concentration of CAMP. So, ang mangyayari po is pag masyado ng maraming CAMP sa loob ng cell, this would push out the water content and also the electrolytes from inside of the cell to the outside. So, mapupunta na lang sila sa intestinal lumen, which is why ito po yung nagpo-produce ng watery diarrhea or yung clinical presentation na watery diarrhea. Kasi nanggaling po yung mga fluid content na yun from inside of the cell. So, parang they only moved outside because of the increased concentration of CAMP. Alright, so that is the mechanism of action of the cholera toxin. Focusing on Vibrio cholerae, this has been well known as the causative agent of Asiatic cholera or epidemic cholera. So the mode of transmission is via the consumption of raw or improperly handled seafood, also dairy products such as milk and unpreserved meat. So medyo marami po yung options, no, as to how we could acquire the infection. So clinical manifestations of Asiatic cholera include diarrhea, vomiting, and what has been described as rice water stool. So yung rice water stool po, this is extremely watery stool with numerous flecks of mucus resembling water from washing rice. Okay? So mukha daw po siyang pinaghugasan ng bigas. Tapos yung mga nakikita po natin na parang yung mga white clumps or particles na nakikita natin dun sa ibabaw, yun po yung flecks of mucus or parang clumps of mucus. Alright, so in untreated cases, constant fluid and electrolyte loss can lead to hypovolemic shock and even death. So dito na po pumapasok yung consequence ng cholera toxin, di ba po? So na-discuss natin at the earlier slide that the cholera toxin is able to cause the accumulation of cyclic adenosine monophosphate, which could push out the fluid content and electrolytes within the host cell. So yun po, parang lumalabas lang siya dun sa intestinal lumen or intestinal tract nung pasyente, and ayan po. So there's fluid loss, dehydration, electrolyte loss, and if the infection is left untreated, this may lead to death. Let's now talk about the next species, which is Vibrio parahemolyticus, the causative agent of what was known as summer diarrhea in Japan. So one of the notable serotype of this organism is the Vibrio parahemolyticus serotype 03K6. So ano po kaya ang meaning ng mga letters na yun? Diba po? The somatic O antigen number 3 and the capsular K antigen number 6. Alright? So that this organism emerged in 1996 and has been linked to many foodborne outbreaks worldwide. So initially it was discovered in Japan, pero ayun po, it was then found out that this organism is apparently present worldwide. Okay, so the mode of transmission is the same as Vibrio cholerae, and this one is a halophilic or salt-loving organism, which would require 1% hanggang 8% sodium chloride concentration for growth. Alright, so dapat po there's at least 1 hanggang as much as 8% sodium chloride concentration dun sa culture media nila. Alright, so still on Vibrio parahemolyticus, after an incubation period of 24 to 48 hours, the patient would now experience watery diarrhea, vomiting, abdominal cramps, and fever, all of which are very nonspecific. So in the laboratory, when isolating Vibrio parahemolyticus, we may be able to observe what is known as the Kanagawa phenomenon. So ma-observe po natin ito kung ang gagamitin natin is the Wagatsuma agar. Alright, so when Vibrio parahemolyticus is plated on a high salt mannitol medium, we would be able to observe what is known as the production of heat stable hemolysin, and the isolates will appear as that of the photo on the right. So notice po, di ba, there are clear zones of hemolysis. So again, this is what is known as the Kanagawa phenomenon. And ma-observe po natin siya using the Wagatsuma agar. So ang contents po ng Wagatsuma agar is there is high salt concentration. It also contains mannitol as a carbohydrate source, and in comparison to SBA or yung sheep blood agar, instead of sheep blood, it contains human erythrocytes. Alright, so ulitin natin, ang components ng Wagatsuma agar are high salt concentration, mannitol as a carbon source, and human RBCs. So again, the Kanagawa phenomenon is the production of Vibrio parahemolyticus of a heat-stable hemolysin, and it would be able to lyse human erythrocytes on Wagatsuma agar. Alright, so on to the next species, we have Vibrio vulnificus. So this has been a well-known causative agent of wound infections and is sometimes referred to as the flesh-eating bacteria. Alright, so ayan po, this picture on the left, ayan po, this is an example of wound infections that may be caused by Vibrio vulnificus. So most likely po, the patient was able to acquire this through occupational exposure. Another is Vibrio alginolyticus. So this can also cause wound infections as well as eye and ear infections, which usually results from also occupational exposure, specifically for fishermen or marine biologists. So, di ba po, sila yung mga usually exposed to those types of environments kung saan pwede nating ma-isolate yung Vibrio species. And sila din po yung directly nag-handle ng mga infected animals from those places, di ba po? Sources of specimen, of course, varies according to clinical manifestation. So, ganun din naman po for other organisms, di ba? If the symptoms are gastrointestinal in nature, the appropriate specimen would be stool. So, for wound infections, swabs are acceptable, and if there would be any delays in processing and the specimen would need to be transported, Carrie-Blair is the appropriate transport media for this case. So recommended culture media to isolate Vibrio species are SBA or sheep blood agar, pwede rin po yung chocolate agar, pwede rin po yung nutrient agar, but with the addition of 0.5% salt concentration. Although the most well-known agar for the isolation and differentiation of Vibrio would be the TCBS or the thiosulfate, citrate, balsol, sucrose agar. Halos lahat ng components ng agar, sinama na nila dun sa pangalan. So let's now talk about TCBS. As shown here on the picture to the left, we may be able to differentiate some of the species of Vibrio based on their ability to ferment sucrose. So on TCBS, sucrose fermenting Vibrio colonies are yellow pigmented, just like Vibrio cholerae, vulnificus, and alginolyticus. So ganito daw po yung itsura ng isang sucrose fermenting Vibrio species on TCBS. So aside from producing yellow colored colonies, nagkakaroon din po ng yellow discoloration sa paligid nila or on the agar itself. Alright, but non-sucrose fermenters such as Vibrio parahemolyticus will appear dark green on TCBS, as you can see right here. Okay, so parang medyo parehas po sila ng konsepto ng McConkey agar, di ba po? On McConkey agar, we may be able to differentiate lactose fermenters versus non-lactose fermenters. So on McConkey agar, yung mga lactose fermenters po would produce pink colonies with a pink precipitate or pink discoloration around each colonies. So, ganun din po dito sa TCBS. Pero ang chine-check natin dito are sucrose fermenters. So, on TCBS, we may be able to observe sucrose fermentation if the colonies produce yellow colored colonies with a yellow discoloration surrounding them. Alright? And ayan po, nakalista na rin po dito yung iba pang mga possible colony morphology nila. Example, on SBA or chocolate agar, they will be able to produce medium to large colonies. They are smooth, opaque, with an iridescent greenish hue. Alright, so if hemolytic po yung strain or yung isolate um, na-collect ninyo, then we may be able to observe their hemolysis on SBA. Kung MAC naman po yung gagamitin, Vibrio species are non-lactose fermenting. So they would most likely produce colorless colonies with no precipitates or discoloration on their surrounding, except for Vibrio vulnificus. So yung Vibrio vulnificus daw po, maybe a lactose fermenter. So, ang Vibrio vulnificus po on MAC would produce the usual pink-colored colonies with pink precipitates on their surrounding. Another laboratory test that we could perform to identify Vibrio as well as differentiate them with Aeromonas species, which is yun na yung susunod natin i-discuss, is the string test. Alright, so this is not to be confused with the Intero string test for Giardia. Okay, so although they have similar names, the procedures and organism of interest is different. So to perform the Vibrio string test, we would mix a small amount of the isolated colonies with 0.5% sodium desoxycholate. So kukuha daw po tayo ng drop ng sodium desoxycholate. So we could either place this on a clean Petri dish or a clean glass slide. So yung drop of sodium desoxycholate plus isolated colonies, pag pinagsama po natin yon, ang mangyayari is cell lysis would occur. So it would lyse the bacterial cells, and this would lead to the release of DNA strands from the organism. And those DNA strands may be observed by pulling the inoculating loop upwards and seeing a viscous string as shown on these photos. So, ayan po. Kaya siya tinawag na string test, no? So, yung nakikita po natin na parang extension or yung string na sumasama or nakadikit dito sa inoculating loop, these are actually strands of DNA that was released by the organism after niyang malyse because of the mixture with sodium desoxycholate. Alright? And ang magpa-positive po dito are the Vibrio species. And magne-negative po dito ang Aeromonas species. Kasi po, mamaya tingnan natin for Aeromonas. Mamaya po kasi malalaman natin that there are a lot of similarities between Aeromonas species and Vibrio species, which is why we have to perform these kinds of tests para po ma-differentiate natin sila. Alright, so again, for the string test, Vibrio will test positive and Aeromonas would test negative. Alright, so let's now talk about Aeromonas. Unlike Vibrio, that has been noted as a curved bacilli, Aeromonas are straight gram-negative rods that are glucose fermenters, and they can usually be found along the same areas that Vibrio may exist. So, pwede rin po tayo makapag-isolate or makapag-acquire ng Aeromonas infections from fresh water, marine water. They're also present among animal meats and produce. So aside from humans, there are a variety of animals that Aeromonas may cause infections on. And so that would include fishes, amphibians, and other mammals. So hindi naman po confined to humans yung infections that may be caused by Aeromonas species. There are two groups of Aeromonas, the mesophilic and psychrophilic groups, and nakadepende po ito on their optimum growth temperature. So the mesophilic Aeromonas thrive at their optimal growth temperature of 37 degrees Celsius, which is approximately the normal body temperature. So there are 3 complexes in this group, and each are composed of multiple other species. So di ba po, yun yung meaning kapag sinabi natin na complex. When we say it's a complex, it does not pertain to just one species. Rather, it's kind of like an umbrella, and under it are a few other different species. So first is the Aeromonas hydrophila complex. So kasama po dito yung Aeromonas hydrophila, bestiarum, and the motile strains of Aeromonas salmonicida. Alright? Emphasis on motile. Alright? Again, ang kasama po sa Aeromonas hydrophila complex are Aeromonas hydrophila, bestiarum, and the motile strains of Aeromonas salmonicida. And then, ayan po, andito rin yung Aeromonas veroniae complex and the Aeromonas caviae complex. Again, all of these are under the mesophilic group. Let's now talk about the psychrophilic group. So, i-compare natin sila sa psychrophilic group. They prefer temperatures ranging from only 22 to 25 degrees Celsius. So, medyo mas malamig ng onte, no? Papunta na po sa room temperature. Alright? So, there is actually only one species in this classification, and it is the non-motile Aeromonas salmonicida. Okay? So, ulitin po natin, no? Ang kasama po sa psychrophilic group is Aeromonas salmonicida, pero yung non-motile strains lang po. So, all the other non-motile variations, serotypes, or biogroups of Aeromonas salmonicida, if there are any, as long as non-motile po sila, dito po sila sa psychrophilic group. Alright? Yung nasa mesophilic group po is yung motile strains. Yung nasa psychrophilic is the non-motile strains ng Aeromonas salmonicida. Alright? We shouldn't really be concerned about this one because Aeromonas salmonicida is not really considered as a human pathogen. Let's now talk about the clinical manifestations that may be caused by Aeromonas species. So, intestinal infections caused by Aeromonas may present itself in 5 different ways. So, first is the very non-specific diarrhea and vomiting. Alright, so this may be confused with any other gastrointestinal infection kasi napaka-basic ng diarrhea and vomiting. Next would be the patient may have dysenteric diarrhea similar to shigellosis with bloody and mucoid stools. So, pwede na po maging kahawig niya yung bacillary dysentery. Third one is the chronic diarrhea, and by chronic, we mean that it can go on for more than 10 days. So, imagine having diarrhea for almost 2 weeks. And then the fourth one is something similar to cholera with the presence of rice water stools. And then the fifth one is the nebulous syndrome or the traveler's diarrhea, which is again something similar to enterotoxigenic E. coli. So in summary, Aeromonas seems to be a multi-talented copycat. No? So parang wala siyang sariling special clinical presentation. It just goes by whatever all the others are doing. So it may be similar to shigellosis, it can be similar to Asiatic cholera, it can be similar to traveler's diarrhea. Right? Do take note though that the most associated complication of Aeromonas infections is the hemolytic uremic syndrome, which is characterized by acute kidney failure and low RBC counts and low platelet counts. Okay, so di ba na-discuss din po natin sa chapter 19, Enterobacteriaceae, yung hemolytic uremic syndrome, which has been associated with EHEC or the enterohemorrhagic E. coli. Alright, so kasama din po sa mga possible complication ng Aeromonas infections yung hemolytic uremic syndrome. Alright, dito naman po tayo sa mga extraintestinal infections. There is a wide range of extraintestinal infections that Aeromonas may cause. But focusing in on wound infections, the most common manifestation of this could start out as cellulitis, but in rare cases, the infection can progress to necrotizing fasciitis, also known as the flesh-eating disease, and also pwede rin po ito magdulot ng ecthyma gangrenosum. So there would be pictures on the next slide. Listed below are common isolates from wound infections. So, pwedeng Aeromonas hydrophila, veroniae, or Schuberti. The picture on the left shows us a case of necrotizing fasciitis. So, as we can see, a large portion of the patient's leg has already been infected, and kung makikita nyo po dito, there are clean, straight cuts. So, most likely po, nagde-breeding na sila sa area na yan. And then this photo on the right shows us how ecthyma gangrenosum would progress. So it would start from A, B, C, hanggang dito po sa D. So it starts off as a very red area at the exact point of inoculation of the organism. And then it would expand and magkakaroon po ng black necrotic center sa gitna. And then eventually, ayan po, ganito yung magiging itsura niya sa letter C. And then, pagdating dito sa letter D, this is how the wound site would heal. So it would obviously leave a very noticeable scar. Let's move along with the laboratory diagnosis of Aeromonas infections. So the most recommended culture media for isolating Aeromonas is the Cephsulidin ergasin novobiacin agar, but with only 4 micrograms of cephsulidin instead of the usual full strength of 15 micrograms. So unang beses po nating napag-usapan yung CIN agar sa discussion natin on Enterobacteriaceae, di ba po? Specifically with Yersinia enterocolitica. So yung Yersinia enterocolitica po is a preferred culture media rin po for the isolation of Yersinia enterocolitica is the Cephsulidin ergasin novobiacin agar, but the original formulation of CIN contains 15 micrograms of cephsulidin. So dito po kung Aeromonas po yung suspected organism natin, we should use the formulation that incorporates only 4 micrograms of cephsulidin. Alright? So, kung ma-conky agar naman po yung ipiplate natin or yung paggagamitan natin for Aeromonas species, we would be able to observe pink lactose fermenting colonies on map. Alright? So, sa CIN po, ayan, on CIN, they are described to have a bullseye appearance due to the center of the colonies being darker than the rest, as we can see here on this top left picture. So again, these are the colonies of Aeromonas species on CIN agar. And then if we were to culture them using BAP or SBA, the colonies listed below will produce beta hemolysis. So yung Aeromonas hydrophila, Veronia, Jandae, and Caviae, ganito po yung magiging itsura nila. So they could produce beta hemolysis on sheep blood agar. Let's now proceed to Campylobacter species. Campylobacter species are microaerophiles, meaning they prefer less amount of oxygen in the incubation environment. So, only about 2 to 10% oxygen ang kailangan lang nila, but the optimal amount would be at 5% oxygen. And then, ayan po, Campylobacter is a zoonotic pathogen that could cause stillbirth or spontaneous abortion among the cattle, sheep, or swine population. So stillbirth or spontaneous abortion, what would happen is they would give birth to babies that are already dead. And then for humans naman po, it causes gastroenteritis. Here is a list of species associated with human infections. So you don't really have to remember all of this, pero ang 3 most common isolates po is Campylobacter fetus, jejuni, and Campylobacter coli. Moving on to the clinical manifestations, specifically for Campylobacter jejuni, the incubation period would take about 2 to 10 days. So symptoms are non-specific and self-limiting. So if we would look at the list of symptoms, ayan po, abdominal pain, diarrhea, cramps, sometimes there would be bloody diarrhea, tapos yung fever, chills, nausea, and vomiting. So again, all of these are quite non-specific. The good thing here is that the infection is self-limiting. So, maaari pong gumaling yung pasyente within 2 to 6 days, even without medical intervention. And po, the problem here is that past infections has been linked to the development of the Guillain-Barré syndrome, or GBS. So, although the Guillain-Barré syndrome is non-communicable, meaning hindi po ito nakakahawa, ano? This is an autoimmune disease. So GBS patients were demonstrated to be positive for antibodies to Campylobacter, meaning the antibodies that were formed during the Campylobacter infection eventually binded to the patient's peripheral nerves, and now this patient's immune system is attacking his or her own peripheral nervous system. Alright? So, parang kaparehas po ito ng konsepto ng mga post-streptococcal sequelae, di ba po, tulad ng rheumatic heart fever and acute glomerulonephritis. So those types of diseases only develop after an infection with streptococcus species. So parang ganun din po, yung mga natira na antibodies from past Campylobacter jejuni infections, those antibodies would eventually bind to the patient's peripheral nerves. And since dun po naipon yung antibodies, iisipin ngayon ng immune system natin na nandun yung kalaban. So our immune system would start attacking the peripheral nerves. So kaya po siya naging autoimmune disorder. Alright, so again, that was the Guillain-Barré syndrome that has been linked to Campylobacter jejuni infections. On to laboratory diagnosis, either stool samples or blood cultures may be collected to isolate Campylobacter, and the preferred culture media for this is the Campylobacter agar. So ang Campylobacter agar po is an enriched selective media that is used in the isolation for this organism, and its components are listed below. So kung makikita po natin, di ba, it contains a Brucella blood agar base. So yung parang primary components ng isang Brucella blood agar, plus meron po itong component na 10% sheep RBCs, and then there's a long list of inhibitory substances such as vancomycin, trimethoprim, polymyxin B, amphotericin B, and cefalothin. So kailangan po natin kailangan natin gumamit ng isang culture media with these inhibitory agents. Kasi po, di ba, yung mga sources of specimen natin such as stool or rectal swabs, these are samples that are non-sterile. So, i-expect na po natin na they contain commensal organisms and other possible contaminants. So, dito na po papasok yung paggamit ng isang culture media na nagko-contain ng inhibitory substances para at least there would be something to inhibit those other organisms, those commensal organisms that most likely are not the causative agents of the disease. Okay, so other suitable media would include the Campi-VCA, the Skiros medium, the Butzler's medium, and the medium V. Alright, so taken from our reference textbook, here are the components of the aforementioned selective media. So, nandito po yung base nila, and nandito rin yung mga additional sources of nutrients. So, example, yung Skiros medium, it contains defibrinated horse RBCs. Tapos, dito naman po sa third column, makikita natin yung listahan ng mga inhibitory agents or antimicrobial agents that are contained within these culture media. Alright, so parang it's the same way on how we studied the selective culture media for Neisseria species, Neisseria gonorrhoeae, di ba po? So, meron din pong iba-ibang culture media for Neisseria gonorrhoeae, and they also contain different antimicrobial agents. So, ganun din po for Campylobacter species. We could use a number of different culture media that also contains variations of different antimicrobials or inhibitory agents. Alright, let's now talk about the incubation conditions of Campylobacter species. So their optimum growth temperature is at 42 degrees Celsius. So aside from 42 degrees Celsius being their optimal growth temperature, this is also the temperature at which most normal flora of the colon are inhibited. So parang dual purpose na po yung pag-incubate ng plates at 42 degrees Celsius. So, maliban sa ito po yung optimum growth temperature ng Campylobacter jejuni, ito rin po yung temperature kung saan hindi na kayang mag-survive ng karamihan ng mga commensal organisms because this would be too warm for them. Di ba po? Pero if our isolate is Campylobacter fetus, then ang recommended incubation temperature pa rin po is the usual 37 degrees Celsius. Okay, now Campylobacter plates must be incubated in both a microphilic and capnophilic setting. So in well-equipped laboratories, this is achieved by making use of the gas pack system created by the BD Diagnostics, and they look like this. So ayan po, this is the gas pack system by BD Diagnostics, and the tiny packets are kind of like sachets at the side of the incubation setup. These tiny packets contain materials that can maintain low oxygen levels and provide the right amount of carbon dioxide that is needed. Alright, so again, ulitin natin, ito po yung gas pack system. And yung tiny packets po dito sa gilid, ito na po yung nagko-contain ng substances na pwedeng magbaba ng incubation or magbaba ng oxygen concentration dun sa incubation setup, and also it could provide the right amount of carbon dioxide. Alright, but here in the Philippines, most laboratories still make use of the cheaper alternative to the gas pack system, and this is the candle jar method, which is literally a candle inside of the jar. So the plates are incubated inside of a jar with a lit candle that is placed on top of them. Alright, so ang principle po nito is the candle would only keep burning as long as may oxygen supply po sa paligid niya, di ba? So if we've closed off the jar, so kung tightly sealed or maayos naman po nating sinara yung jar, what would happen is eventually mauubos din po yung oxygen supply ng candle. So it would eventually burn out. So this would provide the microphilic environment, kasi the candle or the burning of the candle would consume the oxygen inside of the incubation environment. And then at the same time, once the candle dies, ayan po, this would elevate the carbon dioxide level that has been produced due to the burning of the candle wick. So di ba po, kapag namatay yung kandila, magpo-produce ito ng usok. So yung usok po contains the carbon dioxide. So ang mangyayari po is naubos din yung oxygen content and nakapag-produce siya ng capnophilic environment. So that is the concept behind the candle jar method. Alright, so under the microscope, Campylobacter species appear to be curved, and some of them are like long spirals or S shapes, and some of them have even been described to be shaped like seagull wings. Okay, so kapag nakita nyo po yung description na seagull wings, dapat po maisip nyo na agad is the Campylobacter species. So if we would look at this example, ayan po. So mukhang accurate naman. They do kind of look like stretched out letter S's, and ito po, this one or this fourth zoomed in cell, ito po yung tinatawag nila na long spirals. Alright, so ayan, when gram staining, do take note though, that when gram staining, it is recommended that the counter stain be replaced with carbofuchsin for enhanced visibility. So medyo i-modify din po natin yung gram staining procedure para mas maayos nating ma-visualize yung Campylobacter species. Now, if we would like to observe their motility may be observed by performing the hanging drop method. So as shown here in this picture below, they would place a drop of the isolate on a cover slip, and then they would put an O-shaped or circle-shaped ring on top of a glass slide. Alright, so yun po. And then we would put the cover slip on top of that O-shaped ring. So ang mangyayari po, yung drop ng specimen dito sa cover slip, hindi siya didikit or hindi siya mafa-flatten dun, upon contact dun sa glass slide. Kasi po, may nakaharang na ring. Alright? So what would happen is, there's literally a drop that is hanging from the cover slip. Alright? So under the microscope, the bacilli would appear to be darting, similar to like a shooting star when they move. So parang medyo mabilis daw po yung pagdaan nila. Kaya tinawag silang shooting star motility, as we can see right here on this GIF. Alright, so ayun po. All right, so again, that is the darting or the shooting star motility of Campylobacter species. Colonies of Campylobacter appear to be moist and spreading due to the fact that they are motile, but they are non-hemolytic and non-pigmented. So wala tayong ma-observe na color change and wala po tayong ma-observe na hemolysis sa kahit anong culture media na gagamitin natin. Alright, but if you would try to search photos of, or other photos of Campylobacter colonies on the internet, maaari nyo pong ma-observe yung spreading colonies nila. So, it's kind of like a different version of the swarming motility. So, kung ito po yung initial point of growth nung organism, parang merong extension sa paligid nung initial point of growth. So, yun po yung spreading motility or spreading colonies ng Campylobacter species. On to the last bacteria on this segment of our discussion, we have the Helicobacter species. Helicobacter species are the causative agents of gastric, peptic, and duodenal ulcers. So, lahat naman po sila are ulcerations of the gastrointestinal tract. So, nag-iiba-iba lang po yung specific places where they happen. Kaya nag-iiba-iba po yung pangalan. Alright? So, that is for the gastric, peptic, and duodenal ulcers. Helicobacter species has also been associated with the development of gastrointestinal carcinoma. So formerly, type B gastritis was attributed to other causes such as stress or chemical irritants, but later studies were able to show that Helicobacter pylori was in fact the actual causative agent of type B gastritis. Alright, so noong unang panahon po, akala ng mga tao type B gastritis was due to non-infectious causes such as stress, chemical irritants, or foods that the patient may be allergic to. So, ayun po. But, ayan, it was later confirmed that type B gastritis is in fact caused by Helicobacter species. Okay, so let's now talk about this specific species, which is Helicobacter pylori. So gastric biopsy materials may be transported using the Stewart medium if specimen processing will be delayed. So bale, yun po yung preferred specimen natin for this infection, gastric biopsy material. Just as with Campylobacter, the incubation environment must also be capnophilic and microaerophilic. And upon gram staining, they appear as letter C, letter U, or letter S in shape. So, minsan po pwedeng maging kahawig nila yung Campylobacter kapag karamihan sa kanila are S-shaped, as we can see here in this photo. So, ganito po yung typical na itsura ng Helicobacter pylori upon gram staining. So Helicobacter pylori is usually identified using non-culture methods, which is why wala po tayong na-discuss ngayon na colony morphology for Helicobacter pylori. So Helicobacter is a rapid urease producer, and the appropriate media for this is the Christensen's urea medium. Alright, so again, ulitin natin, Helicobacter pylori is identified using non-culture methods. Pero ang basis po ng diagnosis for this is utilizing the fact that they are rapid urease producers. Alright? So, ang gamit po natin is the Christensen's urea medium, and a positive reaction would turn the medium into a bright pink, as we could see here in this picture. Alright? So, ito po yung negative result, or ganito rin po yung itsura ng isang inoculated medium. And then, ito po, this is a weak positive reaction, and this is the actual positive reaction. So, yung buong test media po was turned into a bright magenta or bright pink color. Now, with regards to monitoring patients undergoing therapy, they perform what is known as the urea breath test. So, ayan po. Kasi po, di ba, Helicobacter pylori is a rapid urease producer. So, the more organisms inside of the patient, then there would be more urea that is produced. So, ang mangyayari po if the patient is undergoing therapy, then dapat po bababa din yung number of organisms present in the patient, which would result in less urea that is being produced or less urease reaction that is happening. So yun po yung konsepto ng urea breath test. Here are the references that were used in the making of this presentation. Thank you so much for listening. Let's now continue our discussion to chapter 23. So this presentation contains our discussion on the spirochetes. Here is the outline of our discussion. Just a quick general characteristics on the spirochetes. Spirochetes are slender, flexuous, helically shaped, unicellular bacteria. So although ganyan po yung itsura nila, they are actually just one cell. So they differ from other bacteria that they have a flexible cell wall around which several fibrils are wound. So these fibrils are termed as periplasmic flagella, also known as axial fibrils, axial filaments, endoflagella, and periplasmic fibrils. So ang dami natin pwedeng itawag sa fibrils nila. So I guess for the sake of this discussion, most likely ang lagi ko pong gagamitin na term is the endoflagella, just because this is the one that's easiest to pronounce or say. All right. So ayun po. And this endoflagella is the one that is responsible for motility. So hindi po katulad ng mga kilala na natin na types of flagella, just like the polar flagella, Lophotrichus or Poritricus flagella. Ang endoflagella po is kind of like lodged in between the coils of the cell. It almost looks like it's within the cell. Kaya po siya tinawag na endoflagella, because it doesn't stick out like an actual cell appendage. Okay? And then, also, depending on their species, magkakaiba din po sila ng modes of reproduction. So, Treponema species reproduce via transverse fission, whereas Leptospira and Borrelia divide by the more common binary fission. Let's now proceed with the Leptospira species. Leptospira are tightly coiled, thin spirochetes, and in contrast to both Treponema and Borrelia organisms, the spirals are very close together. So this almost makes them look like a chain of cocci. Ayan po. So electron microscopy reveals a long axial filament covered by a very fine sheath similar to treponines and Borrelia. So on that aspect, magkahawig sila nung iba pang spirochetes. And all species have two periplasmic flagella. Now, for Leptospira species, one or both ends of the organism have hooks rather than tapering off. So as we can see here in this example, parang nakabend or nakahook po yung mga dulo nila. Okay? As we can see right here, this one, so all of them kind of look like that. And then, ayan, their motion is rapid transitional with kind of like a back and forth or rotational movement. So historically, pathogenic organisms were identified as Leptospira interrogans, and the saprophytes were categorized as Leptospira biflexa. And it was then found out that there are about 200 different serotypes of both Leptospira interrogans, sensu-lato, and Leptospira biflexa that are pathogenic. So meron din naman po palang pathogenic serotypes from both groups or species. But again, wag pong kakalimutan that in the past, no? In the past po, Leptospira interrogans sensu-lato were known as the pathogenic species. Tapos saprophytic lang po yung Leptospira biflexa sensu-lato. Now, some serovars of Leptospira interrogans sensu-lato and Leptospira biflexa sensu-lato are pathogenic for a wide range of wild and domestic animals and, of course, humans, but the mechanism of pathogenicity are actually not very well understood. Zoonotic leptospires contaminate water or mud when shed in the urine of infected animals such as rodents, and they often enter the human host through small breaks in the skin or even intact mucosa. Kaya po, di ba, ang pinakaalam natin na paraan kung paano po na-acquire ang leptospirosis ay yung paglusong sa baha. There are two types of leptospirosis. First is the icteric leptospirosis, more commonly known as the whale's disease or whale syndrome. So this is a severe form of leptospirosis that affects the liver and kidneys, and this can cause vascular dysfunction. The other type is known as the anicteric leptospirosis, which is a septicemic form of the infection, causing high fever and intense headache. So the duration of the illness ranges from less than 1 week to up to 3 weeks, and late manifestations can be caused by the host's immunologic response to the infection. So in patients with leptospiral bacteremia, immunoglobulin M or IgM antibodies are detected within the first week after onset of the disease, and they may persist in high titers for many months. Okay? Yung immunoglobulin G naman or IgG antibodies, these are usually detectable 1 month or more after the incubation or after the infection. All right? And then convalescent serum contains protective antibodies. Leptospiremia, or the presence of leptospires in the blood, occurs during the acute phase or first week of the disease, before symptoms are present, which is why toward the end of the first week, as symptoms appear, blood or CSF should be collected. Alright, ulitin po natin yon, during the first week of the infection, kung saan nagsisimula pa lang po na mag-manifest yung mga symptoms, ayan po, ang preferred specimen should be blood or CSF samples. Now, optimal recovery occurs if fresh specimens are inoculated directly onto the laboratory media. Okay? So the best time to collect blood and CSF is during the first week, and dapat po ma-inoculate natin sila agad dun sa laboratory media. So this would lead to optimal recovery. During the second week, urine can also be collected, but the yield is much higher after the first week of illness. Alright? So after the first week of illness, dun lang po tayo makakapag-observe ng a good amount of shedding in the urine. And shedding can occur intermittently for a few more weeks. So kung urine nga po yung na-collect nyo na specimen, then this urine sample should be inoculated immediately onto the appropriate media, because kung papatagalin po natin, the acidity of the urine may be harmful to the spirochetes in the sample. So leptospires are visible by dark field or phase contrast and also immunofluorescent microscopy. So leptospires are obligate aerobes, and they can be isolated in artificial media such as the Fletcher semi-solid medium, the Stewart medium, or the Allinghausen-McCullough-Johnson-Harris semi-solid medium. Okay, so parang halos lahat nung nag-contribute to the development of this culture media is sinama nila yung pangalan nila doon. Allinghausen, Collogge, Harris medium. Imagine may invento po kayong something, tapos lahat kayo nung 10 groupmates niyo, isasama niyo yung pangalan niyo dun sa pangalan ng culture media. Alright? So the tubes are examined weekly for evidence of growth, such as turbidity, haziness, or a ring of growth. So a drop is taken a few millimeters below the surface, and this is examined by dark field microscopy. So we should be able to observe tightly coiled, rapidly motile spirochetes with hooked ends. So ganito po yung magiging itsura nila upon dark field microscopy. Alright, so in patients with leptospiral bacteremia, IgM antibodies are detected within the first week after the onset of illness and may persist in high titers for many months. And then after a month or more, after the onset of illness, IgG antibodies can be detected in some patients. So ito po yung ma-observe natin sa serology. Now, in some laboratories, as shown here in this lower right picture, they have what is known as the Immunoleptospira IgM by the company GenBio, which can demonstrate a good performance in cases of acute leptospirosis, which makes sense, di ba po? Because IgM is most likely detected during the acute phase, which occurs during the first week after the onset of illness. Let's now proceed to the Borrelia species, otherwise known as the blood spirochetes. So mamaya po pagdating natin sa laboratory diagnosis nila, we will find out bakit sila tinawag as the blood spirochetes. The genus Borrelia includes a few different species of spirochetes that are morphologically similar, but they have different pathogenic properties and different host ranges. Kaya po, lahat sila ganito ang itsura upon microscopic examination. Alright, so Borrelia species are highly flexible organisms ranging in length from 3 up to 20 micrometers with approximately 3 to 10 spirals per organism, and they are way less tightly coiled compared to the Leptospira species. Alright, so the diagnosis of borreliosis is done by observing GEMSA or right stained smears of blood samples taken during the febrile period, which is why they are known as the blood spirochetes. Alright, so ito lang po yung mga species of spirochetes na maaari nating ma-observe easily with the help of GEMSA or right staining. As we proceed with our discussion po, you will notice that we won't be discussing them per species, rather we will be discussing them by groups based on what type of disease do they cause. Kasi po, multiple different species can cause the same type of disease, okay? Okay. So let's start off with the agents of European relapsing fever. This includes Borrelia recurrentis and all of its strains, serotypes, or biovars, if any. So these Borrelia species work by evading the complement system by acquiring and displaying suppressive complement regulators. So nagre-release daw po ang mga agents ng European relapsing fever ng substances na pinipigilan yung complement cascade. So when that happens, there would be nothing to enhance the ability of phagocytic cells, the antibodies won't be as effective, and inflammation would be prevented. So all of these are functions of the complement system. So the vector for this disease is the human louse, scientifically known as the Pediculus humanus, as shown here on the photo to the right. Next are the agents of the American relapsing fever, or the tick-borne or endemic fever. So these are Borrelia hermsii, Parkeri, Dutoni, and Turicate. So ang specific vector po nila are soft ticks that belong to the genus Ornithodorus. Let's now talk about relapsing fever. So after the incubation period of 2 to 15 days, massive spirochetemia, or the presence of spirochetes in the blood, would develop, and this remains at varying levels of severity during the entire course of relapsing fever. So the infection is accompanied by sudden high temperature, and there's also rigors or chills and severe headache, muscle pains, and weakness. So the febrile period would last for about 3 to 7 days, and this would end abruptly with the development of an adequate immune response. So imagine niyo po na 3 hanggang 7 days po kayong nilalagnat. So the disease recurs several days to weeks later. So uulit po yung same process na yun after a few days or maybe like a few weeks. So following a less severe but similar course. So dito po nanggaling yung term na relapsing fever because of the recurrence or parang pagkawala at pagbalik ulit ng symptoms ng patient. So the same clinical manifestations may be observed for both the louse-borne epidemic fever and the tick-borne epidemic fever. Next are the agents of Lyme disease, which are Borrelia burgdorferi sensu-lato, Borrelia garinii, Borrelia afzelii, and all their other strains if there are any. So these species are transmitted through the bite of an infected Ixodes tick. So bacterial spread may occur depending on the organism's ability to bind plasminogen and urokinase type plasminogen activators to its surface. So the binding could convert plasminogen into plasmin, which is a potent protease, and this could facilitate tissue invasion, which serves as the virulence factor of the agents of Lyme disease. So another virulence factor is their ability to stimulate pro-inflammatory cytokines in vitro, such as the tumor necrosis factor, which can help in controlling the disease, but also this contributes to the undesirable inflammatory manifestations. Okay. So over here, I've included a bonus video which talks about the life cycle of Ixodes ticks and how they are able to serve as vectors for Lyme disease. So this one is kind of funny. Let's watch it now. Sige. So 10 minutes lang naman po to. It's kind of like science and comedy together. Let's watch this one right now.
Maybe it's happened to you. You're in the shower or getting dressed and you look down and you see a little tick and it's all swollen and engorged with blood. And you freak out and you tell someone, I have a little tick on me and I want your help getting it off. I want you to grab this little tick as close to my skin as possible and pull on it slowly but firmly until it all comes out. And then you take that tick to the doctor and you show them and you ask, is this little tick dangerous? They'll tell you there are two main kinds of ticks, soft ticks and hard ticks, and there are many diseases they can carry. But it is Lyme disease that is the most common and widespread in the world. The ticks responsible for the spread of Lyme are a few species of very similar hard ticks in the genus Ixodes. But before you start throwing blame around, you should know that it's complicated. It's a story with blood, saliva, small rodents, everything you need for a good bedtime read. And here is where it starts. This is Tina, and she is just hatching out of her egg as a tick larva. Right now she looks a bit like demon fingers. Now ticks are arachnids and arachnids have 8 legs, but as a larva Tina only has 6. And she is very tiny, about the size of the head of a pin. At the time of her birth, Tina is completely free of the bacteria that causes Lyme, because mama ticks cannot give it to their babies. So at this point in her life, she could give you a good sucking and you wouldn't get it. Yes, it sounds rude, but it is a sucking, isn't it? Because Tina is a parasite that specializes in eating blood. And she can't help that. Even if she wanted an avocado from Mexico, she couldn't eat it. Not with her crazy ass mouth parts. Sorry, not with her crazy ass mouth parts. We'll get to that in a minute, but first, Jerry, you have to proofread these. But first, how does little Tina acquire the Lyme bacteria in the first place? Or do anything, for that matter? Well, for starters, she will need to eat. Tina will only eat 3 meals in her whole life, which can be a few years long. So the first one has to be a good breakfast. Now, you've probably noticed that she's not much of a sprinter. She also doesn't have any eyes, so she's a bit more of a right place at the right time sort of hunter. Each of her front legs has a special organ called Haller's organ. It can sense moisture, heat, and molecules that help Tina position herself in the likely path of a small animal. If a bird or lizard or rodent passes by, she grabs on. In this case, her first meal is from a mouse. She attaches and starts eating with the crazy mouth parts. We'll get to that in a minute. But what she doesn't realize is that this mouse is what is called a competent host to Borrelia burgdorferi, one of the bacteria that causes Lyme disease. A competent host means that the bacteria can live inside the mouse, avoiding its immune system but without making the mouse sick. Here's what Borrelia burgdorferi looks like. Named after Willy Bergdorfer, by the way. Good name to be named after. You know, you don't want to be named after Eric Diarrhea. No one wants to be Borrelia diarrhea. It is a spirochete, a twisty spaghetti noodle sort of bacterium. Now here's the problem. The bacteria live inside the mouse, but the mouse is going to die at some point. So for the bacteria to live on as a species, it needs to have a way to infect other competent hosts. And this is where Tina comes in. This bacteria has evolved to use ticks exclusively to find new hosts. Tina is sort of like a spaceship that helps it colonize new planets. As Tina eats, the bacteria enters her tummy parts and they attach to the lining of her midgut. Look at that, you can see right through her. Once there, they go into a kind of low-energy state, sort of like those suspended animation pods that astronauts use in science fiction movies. After 3 days of feeding, Tina is about the size of a poppy seed. With her little bacteria travelers on board, she drops back to the ground to digest and to get ready for the next phase of her life. As a nymph, her nymph body develops inside that swollen-ass thing that you really want to pop like a zit. When she emerges, she now has her 4th pair of legs. As a nymph, she will have meal number 2, and this is where it gets interesting. The bacteria are rooting for Tina to pick another competent host so they can infect it and keep multiplying. It's like astronauts hoping to land on an Earth-like planet. But Tina can also feed from incompetent hosts. These include some lizards and all ruminants like deer and cattle. If Tina feeds from one of these incompetent hosts, their blood and immune system will kill off the Lyme bacteria, ridding Tina of the infection. That's right, a deer tick doesn't get Lyme from a deer. This means that depending on her food preference, an infected nymph will either stay infected or be cured. For example, exotic ticks in the southeast of the United States prefer lizards, and therefore Lyme is uncommon. In the northeast, they prefer rodents, and Lyme is everywhere. Now, Tina could also choose a 3rd category of host, which includes people and dogs. In this case, the tick remains infected but gets a good meal. For everyone else, it's a bummer. To the bacteria, we are a dead end. Once inside of us, it has no chance of infecting anything else. And our immune system goes completely batshit crazy trying to deal with it. Anyway, Tina the nymph chooses an uninfected mouse. To see how she transmits the disease, we'll have to look at her crazy-ass mouthparts. Told you, this is what toilet paper looks like in hell. This crazy thing. But let's back up a second. Many other blood feeders, like the mosquito, are quite precise. They poke their mouthparts directly into a blood vessel and let the host's heart pump the blood for them. Ticks, on the other hand, do it differently. They are pool feeders or telmophages. It's sort of like when you're on the beach and you dig a hole and then it fills up with water. Imagine that, but with blood that you then drink. But digging in skin is a little more challenging than digging a hole in the sand. When you're the size of a tick, skin is more like a loose mesh of fibers. Imagine having to tunnel into a giant ball of cotton. To dig, Tina uses these, a pair of chelicerae. Each one can telescopically extend and bend at the tip. By employing a sort of alternate swimming-type motion, they pull the matrix of the skin apart. Tina secretes saliva, which contains proteins that numb the area and suppress the immune system, but also cause tissues to start breaking down. When things have been loosened up a bit, both chelicerae extend, bend at the tips, and pull at the same time. This drives that central barbed thing called the hypostome deeper into the skin. The hypostome seems to act as an anchor, holding on with all those backward-facing barbs. And backward-facing barb is a good pornography name. As fluid from the damaged tissues begin to pool, Tina's mouth sucks it up using a muscular pump. When the first bits of the meal hit the midgut, it seems that the bacteria wake up. Instead of going out through the mouth, which is kind of swimming against the current, the bacteria make their way through the gut wall to the salivary glands, and it's through the saliva that the bacteria enter the host. It can take more than 24 hours of feeding for the bacteria to move to the salivary glands, so if you pull it off early enough, you can avoid the disease. Anyway, after her second meal, Tina is ready for her final molt into her adult form. Males and females look virtually identical in their larval and nymphal stages, but as adults they differ. Females are larger, and males have shorter, stubbier mouthparts. This is because the adult male will not eat again, and instead will use his mouthparts for mating. You'll see. As an adult, Tina will only attach to larger animals because this meal is a big one. Climbing up tall grasses gives her a better shot at big animals. Well now, it seems that Tina has found herself a mate. Here you can see the smaller male position himself to initiate mating. He aligns his jagged mouthparts with the female's genital pore, as is the custom. After some prodding around, and if the vibe is right, he inserts his mouthparts into the genital pore, as is the custom. Well, I know it's blurry, Jerry. You can't hire a good pornographer to film this. It's too small. You need tiny instruments. Now pay attention. While he's doing that, he exudes a sort of liquidy sac called a spermatophore out of his genital opening. You can see it right there. It looks a bit like a white balloon. Then he deposits his sperm into it. You know, you wouldn't hire him to be a birthday clown. When the spermatophore is loaded and ready, he pulls his mouthparts out. Then he pulls the spermatophore over to her genital pore and attaches it. It's like meeting someone who really doesn't understand how to use a condom. Carbon dioxide inside the spermatophore actively pushes the sperm into the female tick. Males can do this 2 to 3 times and often mate with females who are in the process of feeding. It's like a theme restaurant. This last feeding is a doozy. When she detaches after a week or so of feeding, Tina is 200 times her starting weight. This final meal will be used to create her babies. And there's a lot of them. These will be the only eggs that she lays, and they are tiny and vulnerable to dehydration. But she has a special organ called Genet's organ that makes its appearance during the egg-laying process. It looks a bit like a handlebar mustache made out of gummy worms. You can see it right here. As eggs emerge from her ovipositor, Genet's organ catches them between those horns. Genet's horny organ then coats them with a waxy secretion, which helps them cluster together and keeps them moist. Tina will lay up to 2,000 eggs, and when she's done, she will die. Jerry, how long is this clip? Does it just keeps going? Well, no, I know she lays a lot of eggs. I just didn't think we'd watch every single one. I mean, it's less dramatic. And then she will die. That's a fade to black. What do you mean, who eats them? Oh, you mean the ticks? Well, I'll tell you one thing. A chicken will eat a tick. Oh, sure. Chickens will eat ticks all day. Oh, you should see them. Their little heads bobbing up and down. They'd eat a whole bag of ticks if they could. A whole bag of ticks.
Let's take it off like we're on a race.
Alright, okay, so that was the bonus video that I've included, true facts about ixodistic. So it talks more about the life cycle of an ixodistic and kung saan papasok yung part kung saan makikiride yung Borrelia burgdorferi. So ayun po, and paano niya ito matatransmit to the next host. So as explained in the video, ayun po, hindi lahat ng host that the ixodistic would feed on is a suitable host for the Borrelia burgdorferi species. So yung iba sa kanila is the suitable host kung saan patuloy yung na mauulit-ulit yung life cycle nila, and yung iba sa kanila are dead-end hosts, such as us humans. So what would happen is that our immune system would go batshit crazy dealing with them. So that was for the agents of Lyme disease. So now let's talk about the actual disease progression of Lyme disease. So, ayan po, what is Lyme disease? So, Lyme disease is a complex disease that can generally be divided into 3 stages. So, the first stage is localized, with about 60% of all documented cases exhibiting erythema migrans, which is the classic skin lesion normally found at the site of the tick bite. So, this begins as a red macule and would expand to form a large erythema with partial central clearing, sometimes described as like a target or a bullseye lesion, as shown here on the photo to the left. So ito po yung mga bullseye lesion. So this is the area kung saan the ixodistic was able to feed, right? Okay, so stage 2 of the Lyme disease produces widely variable symptoms that include secondary skin lesions, migratory joint and bone pain, and there's also neurologic and cardiac diseases that may develop, plus splenomegaly and severe malaise and fatigue. And then finally, there's the third stage, which occurs mainly in the cardiac, musculoskeletal, and neurologic systems. Arthritis is also a common symptom, occurring weeks to years later. So medyo mabagal din po yung development ng disease. It may take up to years, and in some patients, this has even contributed to the development of Alzheimer's disease. The most common and productive specimen for the laboratory diagnosis of Borrelia burgdorferi sensolata infection is serum for serology. Alright, ulitin natin, ang specimen of choice po dito are serum samples. And this follows a two-tiered approach in which the first step is a sensitive immunofluorescence assay or IFA. And pwede rin naman po yung enzyme immunoassay or EIA as a screening method. So if the test is negative, no further testing is needed, but positive or equivocal results are needed to be confirmed with an IgM or IgG Western blot. So in microscopy, Borrelia species are the only spirochetes that can be visualized with the use of light microscope after GEMSA or Wright staining, which I've already shown kanina po sa general characteristics ng Borrelia species, di ba po? So listed here are the specimens of choice, and this photo on the left is what they would look like kung ang gagamitin naman po natin ay acridine orange using a fluorescence microscope. Let's now proceed to the Treponema species. Pathogenic treponemes are thin, spiral, and approximately 6 to 20 micrometers in length. So they are difficult to visualize with a bright field microscope because they are very thin, but they can be easily seen during dark field microscopy. So basically, yung Borrelia species lang po yung talagang only spirochete that we could easily visualize using a bright field microscope. So the ends of treponemes are pointed and covered with a sheet. So their cells exhibit graceful flexuous movement in liquid described as the corkscrew motility, as we can see here in this GIF. On to the main species that we will be discussing. Let's talk about Treponema pallidum, subspecies pallidum, which is the causative agent of syphilis. So Treponema pallidum, subspecies pallidum, has the ability to cross or pass intact mucous membranes such as the placenta, and they can disseminate throughout the body and infect almost any organ system. So isa po ito sa mga virulence factors niya. Another would be its postulated ability to undergo antigenic variation of cell surface proteins to evade host immune response and establish a persistent infection. So kumbaga, imagine niyo po na kaya niyang mag-disguise at kaya niyang magpalit-palit ng surface antigens para hindi siya makilala ng immune system natin, thereby avoiding immune response. Treponema pallidum, subspecies pallidum, transmission normally occurs through direct sexual contact with an individual who has an active primary or secondary syphilitic lesion. So mamaya pa we'll get to that and learn what we mean when we say active primary or secondary syphilitic lesions. Now, syphilis can also be acquired by non-genital contact with a lesion that may be present anywhere else on the patient's body, or pwede rin naman po through transplacental transmission to a fetus, resulting in congenital syphilis. So congenital syphilis po, this affects many body systems and is therefore severe and mutilating. So early onset congenital syphilis occurs at the age of less than 2 years, and they resemble secondary syphilis in adults, and it is characterized by mucocutaneous lesions plus anemia, hepatosplenomegaly, and CNS involvement. Okay? Kapag late onset congenital syphilis naman po, this results following pregnancies when mothers have chronic untreated infections. So symptoms of late onset congenital syphilis could occur after the age of 2 years, but generally are not apparent until the second decade of their life. So symptoms include interstitial keratitis, and also there are bone and tooth deformities. There's also cranial nerve deafness, neurosyphilis, and other tertiary manifestations. So after bacterial invasion through a break in the epidermis or penetration through intact mucous membranes, the natural course of syphilis can be divided into primary, secondary, early latent, and tertiary stages based on clinical manifestations, but we'll get to that later. There could also be co-infection with human immunodeficiency virus or HIV, and this could result in the variation of the natural course of the disease. Furthermore, the ulcers that are caused by syphilis may contribute to the efficiency of HIV transmission in populations with high rates of both infections. Kaya po, 'di ba, if you remember our discussion on chapter 18, the Haemophilus species, yung lesions or ulcerations that have formed on the patient's genital areas during the course of infection with Haemophilus ducreyi could serve as an entry point for all other sexually transmitted diseases, since sira na nga po yung first line of defense ng pasyente, which is the intact skin. Di ba po? Our skin is considered as a mechanical barrier against pathogenic organisms. So any open wounds or lesions or ulcerations on the skin could result in the entry of pathogenic organisms. Okay, so balik po tayo sa syphilis. Syphilis has a wide variety of clinical manifestations which gave rise to the name the Great Imitator. Let's now proceed with the 4 stages of syphilis. So the primary stage begins after inoculation of the spirochetes into the host, where they multiply rapidly and disseminate to local lymph nodes and other organs via the bloodstream. So parang hematogenous spread po yung mangyayari. So the primary lesion develops 10 to 90 days after infection and is a result of inflammatory response to the infection at the site of inoculation. So the lesion is known as a hard chancre or the Hunterian chancre, and it is typically a single erythematous lesion that is non-tender but firm with a clean surface and raised border, as we can see here on this top left picture. So, ayan po. Raised border, so parang nakaangat or parang namamaga po yung paligid, tapos the middle part is indented or nakalubog yung gitna. Alright? And then this lesion is teeming with treponines and is highly infectious. So, marami daw po na organisms or treponemal cells that is present in this area. So, that makes it highly infectious. So because the chancre is commonly found on the cervix or vaginal wall of women or of female patients, and also this is not tender or not painful, the lesion might not be obvious for these female patients. And the lesion can also be found in the anal canal of both sexes if they practice anal intercourse, and the infection will remain undetected. So for men, the penis is the usual location of the chancre, same as the photo provided. So non-systemic signs or symptoms are evident in the primary stages of the disease, but these lesions are highly infectious. Now, approximately 2 to 12 weeks after the development of the primary lesion, the patient may experience the secondary disease or yung second stage po ng infection, which clinical symptoms would include fever, sore throat, generalized lymphadenopathy. There's also headache, lesions on the mucous membranes, and rashes, as shown here on this bottom left photo. So the rashes can present as macular, papular, follicular, or papulous squamous or pustular, and it can occur on the palms and soles of the feet, as we can see right here in this example. Okay, so all secondary lesions on the skin and mucous membranes are also highly infectious. So katulad din siya ng primary lesion that there are a lot of organisms or treponemal cells that are present on those areas. So the second stage can last for several years or several weeks and can relapse. Okay, ulitin natin. The secondary stage can last for several weeks and this could also relapse. So it might also be mild and go unnoticed by the patient. So again, the symptoms would vary from person to person. After the secondary stage would heal, individuals are not contagious. However, relapses of secondary syphilis could occur in about 25% of untreated patients, and magpa-positive din po sila if they undergo serologic methods of diagnosis. So following the secondary stage, ito na po yung latent syphilis when clinical manifestations are absent. So latency within 1 year of the infection is referred to as early latent syphilis, whereas latency greater than 1 year is the late latent syphilis. So yung late latent syphilis, this is also known as the tertiary stage of syphilis. So symptoms of tertiary syphilis include the development of granulomatous lesions known as gummas in the skin. This could also appear in the bones and the liver. So there would also be degenerative changes in the CNS, which is known as neurosyphilis. There could also be syphilitic cardiovascular lesions, particularly aortitis. There could also be aneurysms and aortic valve insufficiency. Why did that sound so difficult for me? Aortic valve insufficiency. And so patients in the tertiary stage are usually not infectious. So these photos on the left presents to us the physical manifestations of syphilis. Lesions of primary and secondary syphilis typically contain large numbers of spirochetes, di ba po? Which is why nabanggit natin kanina that patients are highly infectious during the primary and secondary stage because a lot of the organisms may be found on those lesions. So the surface of primary or secondary lesions is cleaned with saline and gently abraded with dry, sterile gauze. So, induction of bleeding should be avoided. So, wag pong masyadong forceful sa pag-collect, no? Just apply the right amount of force na sakto lang po, na merong fluids or serous exudates that would come out of the lesions and we could be able to transfer them onto a glass slide. So a cover slip is then added kapag nakapag-collect na po tayo ng transudates, and then the slide is transported immediately to the laboratory where dark field microscopy could be done. Keep in mind though that skin lesions lang po ang specimen of choice natin. Oral lesions should not be examined because numerous non-pathogenic spirochetes may be present and they could lead to misinterpretation of results. Okay, so baka mamaya po, i-report natin as positive for Treponema pallidum subspecies pallidum yung pasyente kahit na iba naman po yung causative agent ng pathogenicity because the sample was contaminated with commensal treponines. So that was for the microscopic methods of diagnosis. Let's now proceed to the serologic methods or serodiagnosis. So serology is the primary method used for the laboratory diagnosis of syphilis. So screening test is done using a non-specific, non-treponemal test. So this detects the presence of non-specific antibodies or antibody-like proteins, referred to as reagent or the Wassermann antibodies. So these antibody-like proteins can bind to either lipoprotein materials from damaged cells or cardiolipin from treponemal cells. So the principle behind this test is if the serum sample from the patient contains Wassermann antibodies, then it should be able to react with the cardiolipin-coated particles, causing them to flocculate. So yun po yung positive result, flocculation, as shown here in this picture. Ayan. So reactive versus non-reactive. Alright, so examples of the most commonly performed non-treponemal screening test for syphilis would include RPR and the VDRL. Next is the confirmatory test, which is a specific treponemal test. So this detects the presence of antibodies to actual treponemal antigens, and there are two methods. First is the indirect fluorescence antibody test, or FTA-ABS. So this procedure involves the overlaying of whole treponemal cells, or treponemes, specifically the Treponema pallidum Nichols strain, to a glass slide, or just a manufacturer created slide, and it is fixed there. And then a heated serum sample from the patient is added onto it. So this should confirm a positive RPR or VDRL test. So kaya po siya tinawag na confirmatory test. So kung nag-positive po yung pasyente sa RPR or VDRL, tuloy po tayo sa FTA-ABS. And a positive result is indicated by the fluorescence of the treponemes kung saan po merong nagbabind na antibody from the patient, just like as shown here on the top left picture. Alright, so ulitin natin, a positive result is indicated by the fluorescence of treponemes. So that was for FTA-ABS. The next specific treponemal test is the T-pallidum particle agglutination test, or TPPA, which makes use of gelatin particles that are sensitized with Treponema pallidum antigens. So a positive result is indicated by the agglutination of anti-treponemal antibodies that are present on the patient's serum. And ganito po yung itsura niyan, as shown here on this lower left photo. On to the last segment of this discussion, let's just quickly go through the other Treponema species. So first is Treponema pallidum subspecies pertenue, which is the causative agent of yaws. So the clinical manifestation of yaws resembles that of syphilis, but the early stage lesions are elevated and they are more like granulomatous nodules. Next is Treponema pallidum subspecies endemicum, which is the causative agent of Behel, otherwise known as the endemic syphilis or non-venereal syphilis. And this is also, this also produces clinical manifestations that closely resembles yaws. So the primary and secondary lesions are usually papules that can often go unnoticed, and they can progress to gummas on the skin, bones, and nasopharynx. Next is Treponema caradium. So Treponema caradium is the causative agent of pinta, which is acquired by person-to-person contact, and this is rarely transmitted through sexual intercourse. So the clinical manifestations are seen as lesions that begin as scaling, painless papules, and then these are followed by an erythematous rash that becomes hypopigmented with time. And then the last one is Treponema denticola and Sokoransky, which are the causative agents of ulcerative gingivitis and chronic periodontitis. So periodontitis po, this is an infection of the gums. Alright, so ito po yung mga species that may be found in the oral cavity of patients. Here are the references that were used in the making of this presentation. Thank you so much po for listening.